Now we want to consider the final level of structure that all proteins exhibit, tertiary structure. Our best approach in this case is to look at multiple examples as well as some common patterns. Let's first remind ourselves what we mean by tertiary structure. It is the three-dimensional arrangement of all atoms in the molecule, its final fold, if you will. We can generally divide the tertiary structure of proteins into two broad categories, globular and fibrous. As illustrated here, fibrous proteins are well named because of their rod-like shape. In this arrangement, the backbone does not fold back on itself, but is rather more extended. The chains of fibrous proteins are parallel along one axis and consist of long fibers or large sheets. They tend to be mechanically strong and for this reason often make up the structural components of cells and tissues. Because of the complex extended network of these proteins, they are insoluble in water. Examples of fibrous proteins include keratin protein, that is a part of hair and wool and skin. Collagen is a fibrous protein that is part of the connective tissue of bones, teeth, and skin. In the figure on the left, you can readily see the extensive network of long collagen fibers. If we look more closely at the structure of collagen, we find that there are three polypeptide chains wrapped around each other in a rope-like twist. This is a coiled-coil interaction resulting from multiple van der Waals interactions. It is known as tropocollagen, with a rather sizable molecular mass. Let's consider the primary structure of collagen and see how it relates to tertiary structure. Each chain of collagen has a repeating sequence of 3 amino acids. The first residue might vary, represented by the letter X. However, the second residue is most often proline or a modified form of proline, hydroxyproline, pictured here, and produced as a post-translational modification. Note that the key feature of the primary structure of collagen is that every third residue is glycine. If we look down the axis of the triple helix, we get a much better idea of how this feature of its primary structure relates to the formation of the triple helix. The position of glycine as the third residue of a repeating sequence means that these residues all lie along one face of a single helix. The triple helix forms from association of these helices in a coiled-coil interaction due to van der Waals interactions. Remember, glycine is the smallest amino acid. Formation of the triple helix could not be so structurally sound if the third residue was any other amino acid. Nothing else would fit within the triple helix. Another factor contributing to the strength of collagen is the hydrogen bonding network involving hydroxyproline and hydroxylysine residues. Over time, collagen helices can be cross-linked by covalent bonds formed between lysine and histidine residues. This is what leads to tough meat in aged animals.